My name is Jessica Torossian. I am a third-year PhD student in Brian Helmuth's lab at Northeastern University. I sort of had a roundabout way of getting here. I started out at NYU. I was majoring in photography, and I actually double majored in philosophy. Then I graduated, worked as a photo editor for a few years before deciding to go back to school. I did my second bachelor's degree in biology at UMass Boston, and then I came here to Northeastern for my PhD. Mussels have been a sort of traditional study organism for the New England rocky shore. Mussels are actually a really important part of the ecosystem. They form these dense beds that provide habitat for a lot of other animals. They stabilize the mud sediments, and one thing that a lot of different people working in this region for lots of years have started to notice is that they've been disappearing from the rocky coast. We're trying to figure out why what we think of, at least in New England, as being a predominantly rocky shore species would be living in these mud beds. They're an important commercial species, and if they disappear, we're not going to have as many to eat. A lot of clammers have noticed that clams and worms and things that are really necessary for the fishery in Maine are disappearing when the mussels disappear. So understanding what they're doing in the wild, as well as understanding their physiology, can help us manage wild populations, as well as a new burgeoning aquaculture industry that's happening here in New England. And one of the things that I'm trying to ask is how are predators affecting how much they need to eat or how their energetics work. So similar to a stress test that you would do on a human, we can do that to a mussel. So we have these really cool infrared sensors that are non-invasive, so I don't actually have to open the mussel up or drill a small hole, which makes it much more likely that I'm going to get a more true heartbeat than something that's caused by the stress of handling them. So we can measure heartbeat rate, respiration rate, feeding rate to see how stressed out they're getting. If we see a tiger in the wild, we might start breathing heavier and get really nervous, and our heartbeat might start going faster. What it's looking like is after a few hours, depending on the size of the mussel and how long that mussel's been starved, we'll actually see that heartbeat rate. You can see the heartbeats, and it actually will start beating much faster after. It usually takes about an hour to see that response, but mussels in the predator treatment do actually start to get a little stressed out by the presence of their snail predators. If there's a lot of predators, those are coming in, they're eating juveniles, they're eating adults, and not only eating them, but if they scare them, that can actually change their ability to survive in the long term. It may mean that they don't eat as much, which may lower their ability to take in nutrients, to take in enough energy to either reproduce or survive in the long term. If you have sites that have very dense predator assemblages, you could maybe see lower survival even if the environmental conditions are actually very good for them to survive.